Welcome to Sciencera. In this video, we're going to discuss the nervous system, chapter number 7. Development and regions of the brain. In the developing brain, the cerebral hemisphere, initially smooth, are forced to grow posteriorly and laterally over the other brain regions by the bone of skull. You can see in diagram A the developing brain of 13 weeks. In adult brain, the cerebral hemisphere, now highly convoluted, enclose the diencephalon and the superior part of the brain stem. The left cerebral hemisphere is drawn so that it looks transparent to reveal the location of deeply situated diencephalon and superior part of the brain stem. In figure B, you can see the adult brain. The brain can be considered in terms of four main parts. Four main parts of brain are cerebral hemisphere, diencephalon, brain stem, and cerebellum. Starting from the cerebral hemisphere. The paired cerebral hemisphere, collectively called the cerebrum, are the most superior part of the brain. The entire surface of the cerebrum exhibit elevated ridges of tissue called gyri, separated by the shallow grooves called sulci. Less numerous are the deeper grooves called fissures which separate large regions of the brain. The cerebral hemisphere are separated by a single deep fissure, longitudinal fissure. Other fissures or sulci divide each cerebral hemisphere into the cerebral lobe. Each cerebral hemisphere has three basic regions: a superficial cortex of gray matter, which looks gray in fresh brain tissue, an internal area of white matter, and the basal nuclei, island of gray matter situated deep within the white matter. Cerebral cortex have four lobes. Cerebral cortex functions are speech, memory, logical and emotional response, consciousness, interpretation of sensation, voluntary movement, and problem solving skills. The first lobe of cerebral cortex is the frontal lobe. Primary motor area located anterior to the central sulcus initiate the voluntary movement of skeletal muscles and motor language too. Premotor areas are located anterior to the primary motor areas, process input regarding to body movement, modifies the movement. Prefrontal area of the frontal lobe, anterior frontal lobe, higher level thoughts, decision making, planning, impulsivity, control, selective attention, personality and problem solving, behavior and emotions. Broca's area is the inferior left frontal gyrus, which control the facial neurons and speech production. Articulation works in the junction with Werkin's area is the language comprehension. Then we have occipital lobe. Occipital lobe is responsible for, it is a visual center. It processes visual information and the visual recognition of the shapes and color. Then we have parietal lobe. The somatic sensory area located posterior to the central sulcus in parietal lobe interprets input from sensory receptor except the specialized senses. It aids in spatial orientation and sensory pathways are crossed pathway. Left sensory areas receive input from the right side of the body. The last one is temporal lobe. This is responsible for auditory processing. Olfactory areas in temporal lobe are located in the deep temporal. Memory, right lobe responsible for visual memory like pictures and faces. Left lobe is responsible for verbal memory like words and names. Then we have functional area of cerebral cortex. You can here see in the picture the motor map in precentral gyrus and the sensory map in postcentral gyrus. One part of the brain is responsible for the motor input and the other is for the sensory input. Sensory areas. Primary sensory cortex makes you aware of a sensation. Association area gives meaning to make association with sensation, and multimodal association area make association between different type of stimuli. This is regarding the sensory area. Then we have motor areas. Motor areas allow you to act upon a sensation. Premotor cortex plans movement, then primary motor cortex sends signal to generate movement. Two special motor cortex are frontal eye fields and Broca's area. Basal nuclei. Although most of the gray matter is in the cerebral cortex, there are several islands of gray matter called basal nuclei, buried deep within the white matter of the cerebral hemisphere. The basal nuclei help regulate the voluntary movement activities by modifying instructions sent to the skeletal muscles by the primary motor cortex. A tight band of projection fiber called the internal capsule passes between the thalamus and the basal nuclei. Then we have diencephalon. The diencephalon are also known as interbrain sits atop of the brain stem and is enclosed by the central hemisphere. The major structure of diencephalons are thalamus, hypothalamus, and epithalamus, as you can see in the diagram. The thalamus, which encloses the shallow third ventricle of the brain, is a relay station or the sensory impulses passing upward to the sensory cortex. As impulse surge through the thalamus, we have a crude recognition of whether the sensation we are about to have been pleasant or unpleasant. It is the neurons of the sensory cortex that localize and interpret the sensation. Then we have hypothalamus and epithalamus. The hypothalamus. It is the floor of diencephalon. Role in regulating body temperature, water balance, and metabolism. The center for many drives and emotions, for example, part of emotional visceral brain. For example, thirst, appetite, sex, pain, pleasure centers in the hypothalamus are all are located in the hypothalamus. Hypothalamus regulates the pituitary gland, an endocrine organ, and produces two hormones of its own. The pituitary gland hangs from the interior floor of the hypothalamus by a slender stalk. The mammillary bodies reflex centers involved in olfaction, the sense of smell. Then we have epithalamus. The epithalamus forms the roof of the third ventricle. Parts of the epithalamus are the pineal gland, which is the part of endocrine system too, and the choroid plexus of the third ventricle. The choroid plexus, which are knots of the capillaries within each of the four ventricles, form the cerebrospinal fluid. Moving on to the brainstem. Brainstem is approximately 3 inches long. Its structures are the midbrain, pons, and medulla oblongata. Provides pathway for ascending and descending tract, has many small gray matter areas. These nuclei produces the rigidly programmed autonomic behavior necessary for survival. Some are also associated with cranial nerves and control vital activities such as breathing and blood pressure. You can see the areas highlighted in yellow are the part of the structure of brainstem, midbrain, followed by pons, and then medulla oblongata. Midbrain. A relatively small part of the brain stem is the midbrain, which extends from mammillary bodies to the pons inferiorly. The cerebral aqueduct are tiny canals that travel through the midbrain, connect the third ventricle of the diencephalon to the fourth ventricular area. Anteriorly, the midbrain is composed primarily of two bulging fiber tracts, the cerebral peduncles, which convey ascending and descending impulse. Dorsally located are the four rounded protrusions called the corpora quadrigemina. These bulging nuclei are reflex center involved with vision and hearing. Then we have pons and medulla oblongata. The pons is the rounded structure that protrudes just below the midbrain. Pons mean bridge, and this areas of the brain stem is mostly fiber tracts, bundle of nerve fibers in the CNS or central nervous system. It has important nuclei involved in control of breathing. Then we have the medulla oblongata. Most inferior part of the brainstem is medulla oblongata, which merges into the spinal cord. The medulla is an important fiber tract area. Pyramidal tract involves motor fiber cross over to the opposite side. Contains nuclei that regulate vital visceral activities. It contains centers that control heart rate, blood pressure, breathing, swallowing, and vomiting, among others. The fourth ventricle lies posterior to the pons and medulla and anterior to the cerebellum. Next is reticular formation. Reticular formation extending the entire length of the brain stem is a diffuse mass of a gray matter, the reticular formation. The neurons of a reticular formations are involved in motor control of the visceral organs, for example, controlling smooth muscles in the digestive tract. A special group of reticular formation neurons, the reticular activating system, plays a role in consciousness and awake or sleep cycle. The RAS or the reticular activating systems also act as the filter for the flood of sensory inputs that streams up the spinal cord and the brain stem. Weak or repetitive signals are filtered out, but unusual or strong impulses do reach consciousness. Damage to this area can result in prolonged consciousness, also known as coma. In the figure, you can see the reticular formation which extend the length of brain stem. Ascending arrows indicate sensory input to the cerebrum. Descending arrow indicates the efferent output of the reticular neuron. Then we have cerebellum. Cerebellum projects dorsally from under the occipital lobe of the cerebrum. Has two hemispheres and a convoluted surface like cerebrum, outer cortex made up of gray matter and an inner region of white matter, provides the precise timing of skeletal muscles activity and control our balance. Fibers each reach the cerebellum from the equilibrium apparatus of the inner ear, the eye, and the proprioceptors of the skeletal muscles and tendons and many other areas. Compare continuously brain's intention with actual body performance by monitoring body position and the amount of tension in various body parts. When needed, the cerebellum sends message to initiate the appropriate corrective measures. Then we have the protection of CNS. The meninges in CNS covers and protect the central nervous system. First one is the dura mater, outermost hard double-layered membrane. It is composed of two layers. Periosteal layer attached to the inner surface and meningeal layer forms the dura mater. The dural layer separates to enclose dural venous sinuses that collects the venous blood. The inner dural membrane folds the falx cerebri and the tentorium cerebelli separate the cerebellum from the cerebrum. Then we have the arachnoid matters. It is a middle meningeal layer and it is a web-like. Arachnoid villi projections of arachnoid membrane protruding through the dura mater. Then we have pia mater. It clings tightly to the surface of brain and spinal cord following every fold. The subarachnoid spaces is filled with cerebrospinal fluids. Let's look at cerebrospinal fluid. Cerebrospinal fluid CSF composition is similar to the blood plasma. It contains less protein and more vitamin C, and its ion composition is different. The choroid plexus continually forms CSF from the blood. The CSF forms a watery cushion that protects and help the brain float so it is not damaged. Inside the CSF is continually move inside the brain, the CSF is continually moving. It circulates from the two lateral ventricle in the cerebral hemisphere into the third ventricle in the diencephalon, and then through the cerebral aqueduct of the midbrain into the fourth ventricle dorsal to the pons and medulla oblongata. Some of the fluids reaching the fourth ventricle simply continues down into the spinal cord, but most of it lates into the subarachnoid space through the three openings, the paired lateral apertures and the median apertures in the walls of the fourth ventricle. You can see in first step the cerebral spinal fluid moving. The CSF returns to the blood in dural venous sinuses through the arachnoid granulation. In this way, CSF is continually replaced. Any significant changes in CSF composition could indicate meningitis or certain other brain pathologies. A procedure called lumbar spinal puncture can obtain a sample of CSF for testing. Then we have blood-brain barrier. This also protects the central nervous system. Neurons are kept separated from the blood-borne substance by the blood-brain barrier composed of least permeable capillaries in the whole body. These capillaries are almost seamlessly bound together by tight junctions all around. Of the water-soluble substance, only water, glucose, and essential amino acid pass easily through the walls of these capillaries. Metabolic waste such as urea, toxins, proteins, and most drugs are prevented from entering the brain tissue. Non-essentials amino acid and potassium ion not only are prevented from entering the brain but also are actively pumped from the brain into the blood across the capillary wall. Although the bulbous of astrocytes or the feet of astrocyte that cling to the capillaries may contribute to the barrier, the relative impermeability of the brain capillaries is most responsible for providing this protection. The blood-brain barrier is virtually useless against fat respiratory gases and other fat-soluble molecules that diffuse easily through all the plasma membrane. Moving on to the brain dysfunctions. The first one is concussion. The slight brain injury causing us to feel dizzy or loss of consciousness briefly. There is no permanent brain damage in concussion. Then we have contusion. Nervous tissue destruction occurs in contusion, does not regenerate. If the tissue is destroyed, it doesn't regenerate. If the cortex is damaged, coma for hours of life can occur. Then we have cerebral edema. This is the swelling of inflammatory response may compress and kill brain cell. Cerebrovascular accidents. Commonly called stroke, the result of a clot or a ruptured blood vessel supplying a region of the brain tissue supplied with oxygen from the blood sources dies. Loss of some functions or death may occur in the cerebrovascular accident or stroke. Then we have Alzheimer's disease. You can see in the diagram the healthy brain, mild Alzheimer diseased brain, and severe Alzheimer's diseased brain. Progressive degenerative brain disease, mostly seen in elderly but may begin in the middle age. Structural changes in brain include abnormal protein deposit and twisted fibers within neurons. Victim experience memory loss, irritability, confusion, and ultimately hallucinations and death. Spinal cord. The cylindrical spinal cord, which is approximately 17 inches, is a glistening white continuation of the brain stem. The spinal cord provides a two-way conduction pathway to and from the brain, and it is major reflex center. Spinal reflexes are completed at this level. Enclosed within the vertebral column, the spinal cord extends from the foramen magnum of the skull to the first or second lumbar vertebra, where it ends just below the rib. Like the brain, the spinal cord is cushioned and protected by meninges. Meningeal covering do not end at the second lumbar vertebra, but instead extend well beyond the end of spinal cord in vertebral canal. Because there is no possibility of damaging the cord beyond L3, or the meningeal sac inferior to that point provides a nearly ideal spot for a lumbar puncture to remove CSF for testing. Collection of spinal nerves at the inferior end of vertebral canal is called the quada equina. Then we have gray matter of spinal cord. The gray matter of spinal cord looks like a butterfly or the letter H in cross section. The two posterior projections are the dorsal horn or the posterior horn. The two anterior projections are the ventral horn or the anterior horn. The gray matter surrounds the central canal to the cord, which contains CSF. Neurons with specific function can be in the gray matter. The dorsal horn contains interneurons. The cell bodies of the sensory neuron whose fibers enter the cord by the dorsal root are found in the enlarged area called the dorsal root ganglion. If the dorsal root or its ganglion is damaged, sensation from the body area served will be lost. The ventral horns of the gray matter contain cell bodies of motor neurons of somatic nervous system which send their axon from out of the ventral root of the cord. The dorsal and ventral roots fuse to form the spinal nerve. Then we have the white matter of spinal cord. White matter of spinal cord is composed of myelinated fiber tracts, some running to the higher centers, some traveling from brain to the cord, and some conducting impulses from one side of the spinal cord to the other. Because of these irregular shape of gray matter, the white matter on each side of the cord is divided into three regions: the dorsal column, lateral column, and the ventral column. Each of the column contains several fibers tract made up of axon with same destination and function. All tract in dorsal columns are ascending tract that carry sensory input to the brain. The lateral and ventral column contain both ascending and descending motor tract. Moving on to the peripheral nervous system. Nerve and ganglia outside the central nervous system. Nerve is the bundle of neuron fibers in peripheral nervous system. Neuron fibers are bundled by a connective tissue sheath. The structure of nerve. A nerve is a bundle of neuron fibers found outside the CNS within a nerve. Neuron fibers or processes are wrapped in protective connective tissue covering. Each fiber is surrounded by a delicate connective tissue sheet and endothelium. Group of fibers are bound by a coarser connective tissue wrapping the perineum to form the fiber bundles or fascicles. Finally, all the fascicles are bound together by a tough fibrous sheet, the epinedium, to form a cord-like nerve, as you can see in the diagram. Nerve that carries impulses towards the CNS are called the sensory or afferent nerves, whereas those that carry only motor fibers are called motor or efferent nerves. Nerve carrying both sensory and motor fibers are called mixed nerves. All spinal nerves are mixed nerves. Then we have cranial nerves. Distribution of cranial nerves. Sensory nerves are shown in blue in the diagram and the motor nerves are in the red. Although cranial nerves 3, 4 and 6 have sensory fiber, these are not shown because the sensory fiber account for only minor part of these nerves. Let's look at each cranial nerve separately. Starting with number one, olfactory nerve. Olfactory nerve origin is fiber arise from the olfactory receptor in the nasal mucosa and synapse with olfactory bulb. The function of olfactory nerve is purely sensory, carries impulses from the sense of smell. Test subject is asked to sniff and identify aromatic substance such as oil or clove or vanilla. Number two is the optic nerve. Fiber arise from the retina of the eye and form the optic nerve. The two optic nerves form the optic chiasma by partial crossover of fiber. The fiber continue to the optic cortex as optic tract. Function of optic nerve is purely sensory again, carries impulse for vision. To test the optic nerve, vision and visual field are tested with an eye chart and by testing the point at which the subject first see an object moving into the visual field. Then we have number 3, oculomotor nerve. Fiber run from the midbrain to the eye. It supplies motor fiber to 4 of the 6 muscles, superior, inferior, medial rectus and inferior oblique, that direct the eyeball to the eyelid and to the internal eye muscles controlling lens shape and the pupil size. Then we have number 4, trochlear nerve. Fiber runs from the midbrain to eye. It supplies motor fiber for 1 external eye muscles, superior oblique muscles. Number 5 is trigeminal nerve. Fiber emerges from the pons and form 3 divisions that run to the face, conduct sensory impulse from skin of the face and mucosa of nose and mouth. Also contain motor fibers that activate the chewing muscles. Number 6 is abducens. Fiber leave pons and run to the eye, supplies motor fiber to the lateral rectus muscles which roll the eye laterally. Number 7 is facial nerve. Fiber leaves the pons and run to the face, activates the muscles of facial expression and the lacrimal and salivary glands, carries sensory impulse from the taste buds of anterior tongue. Then we have vestibulocochlear nerve. Fiber run from the equilibrium and hearing receptor of the inner ear to the brainstem. Function is purely sensory. Vestibular branches transmit impulses from sense of balance and cochlear branch transmit impulse for the sense of hearing. Then we have number 9, glossopharyngeal nerve. Fibers emerge from the medulla and run to the throat. Supplies motor fiber to the pharynx, throat, that promotes swallowing and saliva production. Carries sensory impulses from taste bud of the posterior tongue and from the pressure receptor of carotid artery. Number 10, vagus nerve. Fiber emerges from the medulla and descend into the thorax and abdominal cavity. Fiber carries sensory impulses from and motor impulses to the pharynx, larynx and the abdominal and thoracic viscera. Number 11 is the accessory nerve. Fiber arise from the superior spinal cord C1 to C5 and travel to the muscles of neck and back, mostly motor fibers that activate the sternocleidomastoid and trapezius muscles. Number 12 is hypoglossal nerve. These fibers run from the medulla to tongue. Motor fiber controls tongue movements. Sensory fiber carries the impulse from the tongue. So these were the 12 cranial nerves. Moving on to the spinal nerve plexus. There is a pair of spinal nerve at level of each vertebra for a total of 31 pairs. Spinal nerves are formed by the combination of ventral and dorsal root of the spinal cord. The spinal nerves are named for the region from which they arise. Spinal nerve divides soon after leaving the spinal cord. Dorsal rami serve the skin and muscles of the posterior trunk, and ventral rami forms a complex of network for the interior which serve the motor and sensory needs of the limbs. Autonomic nervous system. Before we move on to the sympathetic division and parasympathetic division anatomy, let's look at the difference between somatic nervous system and autonomic nervous system. Somatic nervous system cell bodies are in the CNS, central nervous system. Axon extend from spinal nerve to the skeletal muscles. In autonomic nervous system contain two motor neurons. Preganglionic neuron in brain and spinal cord. Preganglionic neuron axon leaves CNS to form synapse with second motor neuron in ganglion outside the CNS. Then we have postganglionic axon which extend to the organ itself. Sympathetic division mobilizes the body during extreme situation and parasympathetic division allow us to conserve energy. Anatomy of parasympathetic division. The preganglionic neuron of parasympathetic division are in brain nuclei of several cranial nerves number 3, 7, 9 and 10. The vagus being the most important of these and in the S2 through S4 level of spinal cord. For this reason, parasympathetic division is also known as craniosacral division. The neurons of the cranial region send their axon out in the cranial nerve to serve the head and neck organ. There they synapse with ganglionic motor neuron in the terminal ganglion. From the terminal ganglion, the postganglionic axon extend a short distance to organ it serves. In the sacral region, the preganglionic axon leaves the spinal cord and form the pelvic splenic nerve, also called as the pelvic nerve, which travel to the pelvic cavity. In the pelvic cavity, the preganglionic axon synapse with second motor neuron in terminal ganglia on or close to the organ they serve. Then we have anatomy of sympathetic division. Sympathetic division is also called thoracolumbar division because its preganglionic neuron are in the gray matter of spinal cord from T1 through L2. The preganglionic axon leave the cord in the ventral root, enter the spinal nerve and then pass through a ramus communicans or small communicated branches to enter a sympathetic trunk ganglion. Sympathetic trunk lies along the vertebral column on each side. After it reaches the ganglion, axon may synapse with the second neuron in the sympathetic chain at the same level or a different level, and postganglionic axon then re-enter the spinal nerve to travel to the skin. Or the preganglionic axon may pass through the ganglion without synapsing and form part of the splenic nerve. The splenic nerves travel to the viscera to synapse with ganglionic neuron found in collateral ganglion anterior to the vertebral column. Major collateral ganglia, the celiac and superior and inferior mesenteric ganglia, supply the abdominal and pelvic organ. The postganglionic axon then leaves the collateral ganglion and travel to serve a nearby visceral organ. Let's look at the autonomic functioning. We're going to look at the parasympathetic division and the sympathetic division separately. Starting with parasympathetic division, it is housekeeping activity, conserve energy, maintain daily necessary body function, also known as D division, because responsible for digestion, defecation, and diuresis. Pupil constrict, it conserve energy, stimulate digestive system, increase saliva, stimulate urinary hunger by increasing the urine production, inhibit cardiovascular system, slows heart rate, inhibit respiratory system, constrict airway or slows respiration. Sympathetic division, also known as fight or flight mode, responds to unusual stimulus take over to increase the activities. Also known as E division, responsible for exercise, excitement, emergency, and embarrassment. Pupil dilates, stimulate respiratory organ by increasing breathing rate and open airways, stimulate cardiovascular system by increasing the heart rate, stimulate adrenal gland by release of epinephrine and non-epinephrine which further stimulate the liver. Stimulated liver convert glycogen into glucose and it is released into the blood. Inhibit digestive system by decreasing the saliva and causing dry mouth, and inhibit urinary system by increasing glucose use which generates heat and water by increasing sweat and the skin is reddened. Development aspect of nervous system. Nervous system is formed during the first month of embryonic development. There are no more neurons formed after the but growth and maturation continues for the several years. Neuron will develop myelination during maturation, increasing the neuromuscular control. Brain reaches maximum weight as a young adult. Maternal and environmental factor may impair embryonic brain development, like oxygen deprivation destroy the brain cell. Severe congenital brain disease include cerebral palsy, anencephaly, hydrocephalus, and spina bifida. Premature babies have trouble regulating body temperature because the hypothalamus is one of the last brain areas to mature prenatally. Development of motor control indicates the progressive myelination and maturation of a child's nervous system. Brain growth ends in young adulthood. Neurons die throughout life and are not replaced, thus, brain mass declines with age. Healthy aged people maintain nearly optimal intellectual function. Disease, particularly cardiovascular disease, is the major cause of declining mental function with age. In the diagram, you can see an embryo with 1 month old developing forebrain, midbrain, and hindbrain. Embryo of 5 week old develop telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. Brain structure present in adults: cerebrum, including cerebral hemisphere, in cerebral cortex, white matter, and basal ganglia develop. Diencephalon, like structures like thalamus, hypothalamus, epithalamus develop. Midbrain form the part of brainstem. Pons, also a part of brainstem. Cerebellum develop. Medulla oblongata, part of the brainstem, also present in the adult brain. This brings us to the end of nervous system. Thank you for watching. If you like my video, don't forget to subscribe for more.